Excellent. Well, everybody, welcome. We have a webinar today on lung tissue and PCLS given by Dr. Anne van der Does. She is with the Department of Pulmonology in Leiden. So Dr. Anne van der Does is an assistant professor in the Department of Pulmonology at the Leiden University Medical Center in the Netherlands. Her work is focused on dysfunctional lung epithelial biology in chronic lung disease, with specific focuses on developing advanced lung epithelial cell cultures to support that research. And she's going to talk about a chip today. And related to this focus, Dr. van der Does was awarded actually the Marie Curie Fellowship twice, of which the second included a one-year visit at Emulate Incorporated, which is a pioneering company in organs on chips technology, and she is helping develop their Lung-on-Chip platform. She has published in many peer-reviewed journals, including the European Respiratory Journal. And so today, she's going to share with us her research on how to leverage primary lung tissue to develop this cutting edge, I like the pun, lung cell culture models or chip technology. So with that, Dr. van der Does, I want to hand it over to you.
Well, thank you very much. Thank you very much for the invitation to present my work here in this webinar, but also thank you very much for the kind invitation, for the introduction. I wanted to start off with saying that this work that I will present today is really a shared work. So as you already mentioned, I received this Marie Curie, in which I included a visit to this company, Emulate, and that's the platform I will talk about today, about the Airway on-Chip that we that I worked on when I was visiting them. And then I got to know Dr. Jana Norrold, and together with several students that were present at Emulate that now work either in Jana's lab or moved on to with their careers to PhDs. And so they all contributed to this work. And in the end, of course, of my slides, I will acknowledge everyone individually, but I think it's really nice to know that this was really a shared effort between different partners here. So that's really nice. So indeed, I work in the Netherlands in the Pulmoscience lab, and basically in our lab we work with in vitro culture models. And the question is always like, why would you want to only study lung diseases in vitro? In this particular journal or manuscript, it was a good case was made why mice are a good model for human airway diseases. But in that same journal edition, there was also a case made why mice are not such a good model of human airway diseases. And actually, this has already been published over 20 years ago, and this discussion have been ongoing. But if you look actually at the data, you see that there's not such a great translational value of animals when you look at how many products that are actually tested in mice that work well actually do not work in humans and don't make it to the clinic. The predictive value is not very great, and there are several reasons for this. And when you look into the lungs specifically, the biology and the anatomy is actually quite different between mice and human. For example, the epithelial lining has a different cellular composition in different ratios in the different regions of the lungs of those that compare to humans, because also not all regions are represented. And if you look at the anatomy, the bifurcation is very different. So the angles are different. So that means that the deposition of particles, for example, is different. And altogether, this probably contributes to why we are convinced that if we improve lung models, in vitro models on a human background, it hopefully increases that's what we assume, increases the predictive value, and we can transition into research where we don't need these animals anymore, mostly for the ethical reasons, obviously, and also because, as I mentioned, the predictive value needs to be increased. So the platforms that we use in our lab are all based on human primary epithelial cells that we have a biobank of, from the nasal region up to the alveolar region, and also endothelial cells and some other lung tissue-derived cells. We also use iPSC-derived alveolar epithelial cells, but I will not address that today. And then the platforms that we culture these cells in are various. So we have the more golden standard, let's say traditional insert cultures, the air-liquid interface, where we create an epithelium on top of a membrane that we first submerge, but later culture at the air-liquid interface. And we can expose these cultures, for example, to whole cigarette smoke or bacteria or viruses. We also use organoids, but organoids we mostly use for sort of prolonging the expansion of the primary epithelial cells. As I said, we do a lot of exposures in our research, and organoids are not such an ideal platform for that. Although we do other experiments as well, for example, radiation-induced lung damage, we investigate using organoids. We also have various lung-on-chip models. So the commercial lung-on-chip model that we have from Emulate, that's what I'm going to discuss today. But we also recently developed an in-house model where we have a vascularized hydrogel in which we can study the interaction of the vasculature with the epithelial airway or alveolar epithelial compartment. And finally, we also use some PCLS in our cultures. Because of the background of this webinar, you would expect that I would talk about that today. But I am actually not doing that. I'm going to talk about our lung chips because we use the machine, the compressed tome microtome, to sort of use it as a tool to get a better imaging of the side view of the chip. And I thought it would be quite an original angle to not talk about PCLS, but actually a different way we use the machine in this particular research. So when we use this platform, we instantly, let's see if my slide wants to progress. No. Let's see. Here we go. So when we do the when we use our platforms, we basically always have the stress of what region do we want to represent. When you look at the for example, the epithelial lining in the airways, when you descend from the trachea into the lungs, it changes. The cellular composition of the epithelial cells changes. And of course, single-cell RNA sequencing showed a lot of different epithelial cell types, many more than we used to think they were there. But on the upper right corner, you can see the presentation of the, say, the most dominant cell types in the airway epithelium. And if you just look at those ones already, they change a lot when you go deeper into the lungs. So where you have in, say, the tracheal and the large airways, a little bit more stiffer surrounding because it's supported by cartilage. We have a lot of basal cells and goblet cells. When you go deeper into the lungs, this cartilage disappears, the basal cells become lower in number and the goblet cells as well, while the club cells, for example, increase in number. And when you reach the alveolar space, everything changes. There is basically the maximum amount of stretch related to breathing over there. There's different epithelial cell types because, of course, the function is different in this part of the lungs. The gas exchange takes place, and you have different epithelial type 2 and type 1 cells. So at this point, we still really have to choose which region we want to represent in culture. And besides that, not just the biology changes throughout the lungs, also the mechanobiology of the lungs. So the stiffness, the stretch, but also the airflow velocity. And when we were looking into these changes and thinking about these regions, we actually realized that the gradient that you see in the changes of the airway epithelial composition are also aligning the same gradient where you see these differences in stretch and the airflow velocity. And we wondered if these features were perhaps connected. So we were wondering, how does airflow and stretch and mostly the changes therein affect airway epithelial functioning? When we got interested into this research question, the only commercial organ-on-chip platform that was available that was already that advanced was from Emulate. We wrote this grant together, and so I could visit them to learn to work with this chip. And basically their platform is comprising of several components. So it's all, of course, evolves around the chip, which you can see here in the bottom right. It's actually integrated in this gray carrier, and it's basically the size of a small USB stick, or a traditional USB stick, I should say. And in the chip, we culture the cells, and then once the cells are adhered to the membrane, the chip is integrated in the pod, and this pod is basically a medium reservoir, and you put the medium in the reservoir, and then it's pushed through the chip, through the channels, and then it enters the waste reservoir on the other side. So it's basically a flowing system, and when you put this chip and pod together, it's a sterile circuit, so you could look at the cells under the microscope, for example, and basically work with it without getting the cells infected. So you can then place this unit, the chip pod unit, into Zoe, and the Zoe basically you can program to have different flow rates or different stretch and the amount of stretch, and also the frequency of the stretch. So with those cues, you can actually mimic the breathing, let's say, forces that you also have in the lung. And we wanted to really use that, leverage that part of the system to investigate how it affects airway epithelial biology and functioning. The Zoe is placed inside the incubator, as you can see over here, and the orb is placed next to the incubator, and that basically regulates the stretch and gets the Zoe to do the things that the Zoe does. So zooming in a little bit more on the chip, how does this look like? Oh, this is going to be a problem. I don't understand, but it's not able to play the media. Well, I have a lot of videos in here, so I really hope that the rest of the videos work out. I think it's probably something with Zoom. So Abby, if you have any, let me know.
Otherwise, I'm not sure, Dr. Vander Duss.
Please
I
say
don't
Anne,
know
it's
if you
fine.
have the videos. You can always exit out of your PowerPoint screen to show them. But I'm not sure.
No, me neither, because I normally have.
If you click the play
Teams
button, what happens?
Yeah. I don't think it works, but let's see if the other videos work or whether it's just this video. Basically, what I wanted to show here is that the chip contains of two channels that are on top of each other, separated by a membrane, and this membrane is flexible, so it has large pores. So technically, you could have cells migrating from one side to the other, for example, immune cells, which were already published about. And then in the top channel, we represent the airway lumen. So that's where we culture our airway epithelial cells. And in the bottom channel, you could integrate vascular endothelial cells, for example, to represent, say, the bloodstream. In our study, we only used airway epithelial cells, but I'll come back to that later, because we really wanted to see how the biomechanical cues would actually affect the airway epithelial cells, so we didn't want to put any other cell types in there. And on the side of those two channels, there are two empty channels, and those two empty channels can be put on a vacuum, and then they shrink, and they basically, because of that, pull the membrane apart, and that's how you create the stretch in the chip and these breathing-like motions. Let's move forward. So basically, the setup of our study was like this. It's a bit of a complex, but I'll talk you through. So this is, again, this is sort of a cross section of the chip, where we have in our study only medium. So in this case, this is an optional. We did optimize or develop the protocol that you could also integrate endothelial cells. But in our study, we did not have endothelial cells there. So basically, we had medium in the bottom channel. And in the top channel, we cultured airway epithelial cells that we differentiated at the air-liquid interface. And what we did is we compared traditional inserts in which we differentiated the airway epithelium as well for 14 days on the air-liquid interface. We did the same in the control chips, but then with perfusion of the bottom channel, so with perfusion of constantly new media. And then in some of those chips, we expose these after 1 week of air-liquid interface. We wanted to give the epithelium a little bit of time to create tight junctions and to create a barrier. We weren't entirely sure how it would affect them, of course. So we thought this would be the best setup. So we differentiate them similarly to the control chips for 7 days at the air-liquid interface. But then for the second 7 days, we expose them to airflow and stretch. We gave them 5% strain. It's very difficult to establish the amount of strain in the airways, but this should sort of resemble the smaller airway region, the stretch that you could expect there. And also with 0.25 hertz, we had a normal breathing as frequency. So again, just to, because this sometimes is a bit complex, so the insert and the control chip were basically cultured the same. So on air-liquid interface for 14 days, except that in the control chip, of course, we had the medium flowing. The air is static in this chip, but in the airflow and stretch chip, we had airflows flowing and stretch applied to these chips. And this is for the final week of differentiation. So then first we needed to optimize the airway epithelial cells in culture because we were used to culturing the cells on inserts, and this is a very different membrane. It's a stiff membrane from PET, while in the chips there's a more softer membrane, if you will, from PDMS. And that needed us to change somewhat the coating and the culture conditions. Let's see. Yeah, so that worked out well. And yeah, I'm very sorry that I cannot play any of my media. I don't really understand. But I have these beautiful videos where you can really look into the chip very well. And you can see here the top channel where you have the airway epithelial cells, in sort of green, you can see them now. In this particular chip, because we wanted to show all the different channels, here are the endothelial cells in the bottom channel. But again, in the studies that I'm going to show, we did not put any endothelial cells in there. But here you can see them. I can unfortunately not see how nice we can image those. Let's see if we can move forward. No, it seems that's really struggling to. Here we go. So then we also wanted to, because as you can see over here already, that even though you have a side section via Z-stacks on the normal microscope, we did not have any resolution where we could actually see the cells. But when we had the cells differentiated on the chip, we really wanted to make sure it was a normal airway epithelium, such you have in your own lungs, where you have a pseudostratified layer, where you have basal cells on the, say, the basal side of the epithelium, and the cilia, of course, on the apical side of the epithelium. And this is what we wanted to have. Like, we wanted to have a cross-section where we could really see that the epithelium was well differentiated and the cells were attached to the membrane. And to get that cell resolution, we had to zoom in really to get into the side view. So then, after thinking about how we could reach this, and we tried several things, a PhD student of us suggested to try to use the PCLS machine for this, and our technician has optimized this method. Unfortunately, the media don't play, so I cannot show you this, but maybe afterwards I can switch my screen and maybe show you some videos. But basically what we do is we cut away the excess PDMS, so we really cut out, let's say, the channel part of the chip. Then we glue on the piston from the machine, we put a little bit of super glue, and then we put the chip, or what is left of the chip, if you on top of this pistons until it's solid attached, and then we integrate it in the machine. And then let's see, here we go. And then we have the machine, of course, cutting the way it always does, and then we have this small slice of chip that we can take off. And then here you can see from a top view that little piece in a well. That's how we fix them, or they're already fixed, but that's how we stain them. And then we can actually image these, and we can always, it's also easy to take the chip away again by cutting off the chip from the piston. It's very easy, so you can reuse it. You don't have to order a new piston every time you want to do this. And actually we could then get this resolution. So from the side view, you can now see that the basal cells that are expressing keratin 5 are at the basal side of the epithelium. So you're underneath, you have the membrane. The non-basal cells, which express keratin 8, are all green here. And on top, you can see the cilia, the hair-like structures from the ciliated cells that are positive for acetylated alpha tubulin. And here you see a bright field image, so you could also look at thickness, for example. And this is an H&E staining. That was one of our tries to see if you can image it well. So you can really see that this way we can get a good resolution. It is sometimes tricky to get, so if you really want to have this type of resolution, you need to have quite thin slices, and sometimes the epithelium detaches from the membrane when you cut, so you have to optimize a little bit yourself how thick you need to cut the slices, and you have to probably try a few times before you have a nice slice to image, but it works quite well. So we're very happy that we can use the machine now for a different application. All right, so what we did then was we wanted to figure out what airflow and stretch, but also how the chip culture on itself compared to the insert, what was happening with these cultures and if they had any differences. So what we did is, because we could make some educated guesses, of course, but we thought it would be better to do an RNA sequencing, bulk RNA sequencing analysis, and let the data guide us on the biggest differences. And actually from the gene set, it was significantly different between chips and inserts. One of the dominant pathways came out was tissue development. So that really sparked our curiosity. So tissue development, we really thought immediately, oh, this must be something with maybe accelerated differentiation or possibly different in mucociliary functioning. So what we did first is we characterized the ciliary activity and also its functioning. So again, this is a video of really nicely beating cilia. You could really see all the cilia beating, and what we can do with that, with those videos. And this is really Jana Narwold's expertise. She can use these videos to determine the ciliary beat frequency and the ciliary density. And when we did this for the chip and we compared those with the insert cultures, we actually found no difference. We found that there was similar amounts of ciliated cells being present, and also that the ciliary beating frequency was also the same. But then, so that's summarized here. But then what was interesting is that the mucus clearance, and again, this is a video where we basically what we add is a solution with fluorescent particles on top of the cell layer. And the cilia, they start beating, of course, and then we can track over time, it's a 20 seconds videos, we can track the displacements of the beats, the fluorescent beats, and those that we can then project into an image that you can see on the right side. And then you can follow those trajectories and you can see how much fluid was displaced and with what speed and how, so it's basically how well the cilia are functioning. And what we could see was quite striking, that despite the same amount of cilia present and that they were all beating similarly to the insert cultures, the chip cultures actually showed that they had a lot more area that they could displace the beats over, but also that the speed was, of course, related then much quicker. So the inserts at day 14 days after air-liquid interface, despite the presence of all these cilia, they were not so functional yet. They couldn't displace any fluid yet, which you can see over here. But on the chip, this was already done well. So just to exclude that there was something wrong with the inserts, it's also published that insert cultures take a longer time to completely mature with regard to their mucociliary clearance. And indeed, when we left the cultures for longer, so these inserts were cultured from day 14 up to day 35, we saw that this mucociliary clearance was completely present after 35, while still absent at day 14. But it seemed that the chips were much quicker already there, so that they seem to have an accelerated maturation of the mucociliary function of the airway epithelium when cells were cultured in these perfused chips. So then, of course, we were interested in what happens then if you add airflow and stretch to these chips. And then another interesting thing we found, basically, so the displacements of the beats by the cilia was in one direction. But when we applied airflow and stretch for a week to these cultures, we saw that the directionality was much more polarized compared to the chips without airflow and stretch. So basically, it was more directed to the same area in the airflow and stretch exposed chip, and this was significantly improved in these chips. So this exposure clearly then added another layer of this mucociliary maturation, accelerated mucociliary maturation. So then we thought maybe something is different with the cilia. So let's go back to this RNA-seq data set and see if we see any strange things happening in cilia-associated genes. And actually, when we looked at the sort of structural genes, nothing popped out. We also did some extra qPCRs to verify if some of these major genes with when we used more donors would change, but we did not find any differences. What we did find is that the major PCPs, the planar cell polarity gene and also protein, showed differences. So on gene expression, we saw a slight increase in chip cultures compared to the insert cultures, but what's more strikingly on the protein level, and that's where this is different, is most important, is that what you can see is that in the chip cultures, if you stain for this VANGL1, planar cell polarity protein, which is really connected to the maturation of cilia, you can see that they form this crescent ring around the outer side of the cell. While in the insert cultures, it's still very diffuse throughout the cell expressed. And especially this crescent is a key indicator of functional maturation of the ciliary beat movement. And this confirmed basically that the maturation of these cilia was accelerated in chips by just already in the chip culture, and that was even more accelerated or more even better with airflow and stretch application. So then we also looked at the cellular differentiation. So did the cell composition also change? So we already saw that there was not so much effect on the ciliated cells. Well, indeed, also on the other differentiated cell types, so club cells, goblet cells, and ciliated cells all did not show any difference between insert cultures and chip cultures. We did see some difference on the basal cells, but again, in the specialized cells, there was no difference to be found between chip or insert cultures. When we compared airflow and stretch on this right side, we did not find any major difference, except that we found that after airflow and stretch application, there were more club cells, or at least the gene expression of the gene related to club cells was increased, but we could not confirm this on protein level, so we're not entirely sure if this means anything. But overall, we could not see massive changes in cell composition in chip cultures compared to insert cultures, or chips that were exposed to airflow and stretch compared to control chips. Some other things that we found in our data set, or by doing extra experiments, is that we did find sort of an effect on of the airflow and stretch application on the baseline levels of some pro-inflammatory markers, such as IL-8. This is a protein level of IL-8 that you can see that is decreased in the air. This is the bottom line is airflow and stretch exposed chips. So over day of culturing on airflow and stretch, so the second week of differentiation, we see that it's significantly lower, especially in the later on compared to the control chips, and also COX-2, or this shows the gene for COX-2, PTGS2, was significantly reduced, which is an inflammatory gene in airflow and stretch exposed chips. We did not see major effects on ECM-related genes, but some of them did show changes. For example, MMP9 and fibronectin, the genes encoding for those proteins, were significantly reduced in the genes related to airflow and stretch. And this is something that we find very interesting, especially in relation to, for example, lung fibrosis. So that's something we really want to investigate further in the future. So then, in conclusion for this story, is that we basically have compared traditional insert cultures with organ-on-chip cultures or perfused cultures, where we did airway epithelial cultures for 14 days at the air-liquid interface. And what we found is that if you take the traditional insert culture with static medium and static air, we found that we got, after 14 days, a good proportion of ciliated cells that showed normal ciliary beating frequency, but had the absence of mucociliary clearance and planar cell polarity protein expression inside this crescent shape. But when we culture these cells, the same cells, then on the organ-on-chip platform, with the medium flow during the differentiation, we see that again we have similar amounts of ciliated cells and ciliary beating frequency, but we also see that the mucociliary clearance is present, although the planar cell polarity could still be better. And when we then add airflow and stretch to these cultures during the final week of differentiation, we see that we have the ciliary cells present, the ciliary beating frequency is comparable, the mucociliary clearance is very good, and also the planar polarity or the maturation of the cilia is also confirmed. So then just the final, and we can use the machine now to also look to confirm if the differentiation is on the cellular level is pseudostratified. So then the final slide that I wanted to show you is that we are going to also use the machine for PCLS, of course. So we have a very cool study where we can combine the machine basically for using it for PCLS, but also for the chip, because this project combines tissue analysis and also culture on the chip. We're very interested in modeling the COVID-19 associated pulmonary tissue damage, mostly sort of the fibrotic phenotype that you see in patients that were admitted to the hospital with severe COVID-19 and were admitted to the ICU, and some of those patients received mechanical ventilation, and mechanical ventilation is a risk factor for this fibrotic damage, fibrosis damage in the lungs. And we really want to understand that a bit better, and also why in some of these patients the tissue damage is repaired again, while in other patients this is not. And so what we really want to do is we want to infect, we want to recreate this environment. So we want to infect tissue slices with SARS-CoV-2 high levels, and understand then the process of recreating this, let's say, severe lung damage. And then the key mediators in that process we want to transfer to the chip, where we want to co-culture. We already have done some pilot work, so we want to co-culture the alveolar epithelium together with macrophages. So far, we can see here on the bottom you can see that these are macrophages that were fluorescently labeled, because with the bright field image that you see on the left, you cannot discriminate the alveolar cells well from the other cells. So these are alveolar cells that are with macrophages cultured on top. And then in this particular study, we put a fibroblast in the bottom channel, but we likely will change this when we have more information from the tissue slices. And then we can apply different levels of stretch to the culture and understand how, say, more physiological levels or more pathological levels perhaps affect this fibrotic process. And we hope that we can then identify interesting pathways that we can study further, mostly, of course, to promote repair in the future. So having all this said, I would really like to thank and acknowledge the people that were involved in this. So as I said, we all met at Emulate basically. So Jana Nalroth was working there still, but now she has her own lab in Germany. And Doris Roth, who was also at the time present, works now for her as a PhD student. And we still work together a lot, also finalizing this study. There were people from the core facility in this in the LEMC here of the imaging core facility that helped out with all the imaging, the lovely videos that I unfortunately couldn't show. Then, of course, I want to thank the Ryan Firth lab from, originally the University of South California, but now she has her lab at the University of Iowa, and Christiana from her lab that was involved in this study. And finally, of course, Emulate, who contributed throughout the study with being connected to the project, of course, via the Marie Curie action. And of course, all the funders that contributed to this project. So thank you very much. And if you have any questions, I will be happy to answer them.
Well, Dr. Vander Dues, thank you so much. It's really exciting not just to see how you have different applications for the instruments, but also to see that your research is headed towards understanding COVID-19 more, because it's so applicable. I want to open the floor up now and see if anybody has any questions. I didn't receive any in the chat box, but please feel free to enter now or unmute yourselves and also ask questions if you like. I just want to give a moment and open up the floor. And please also afterwards, if you come up with questions later on or want to share or ask anything, please feel free to reach out to me directly and I'll put you in touch.
Yeah, please also, I forgot to say, you can also email me directly if you have any questions or interest, or you can look at our website to see what we use this chip more for, and or follow us on Twitter. We regularly-ish post.
Well, we have a very nice comment from Dr. Ricardo Pineda, who we've worked long with for tissue slicing. He just said very nice system. We did have a one question, which is normally differentiation takes 3 to 4 weeks for the airway. Can you let us know how you achieved it within, I believe this person said 21 days rather than 21 weeks, Sasha, is that right?
Yeah, so maybe I can, so in our traditional insert cultures in our lab, we also always use the cells already after 14 days of air liquid interface. All the major cell types are then expressed already. We actually found out through this study, because we haven't never been able to analyze this before, but that was because of the expertise of Jana, that the mucociliary displacement is so that they're not completely matured yet with regard to mucociliary clearance. But generally, in the way we culture ourselves, and this is I would say in the lung chip even accelerated, they are already representing all the major cell types after 14 days of air liquid interface culture.
Got it.
And I think it's probably influenced by the way you work up the donor. So the donor material that you get, it's donor dependent somewhat, although, yeah, I would say in our cases, we always use, for every donor, it works out at least 14 days of air liquid interface. But it's also the pre-work, like how many times do you expand them before you put them in your biobank, for example, the type of media likely that you use. We know that coating can make a difference. Although we have to say we used 2 different coatings for the insert, because we had to change our coating for the chip, and we weren't sure if the inserts would be comparable if we kept them on our traditional coating. So we used two coatings, the new one and the old one, but those actually turned out to be comparable in the insert. Yeah, so I think it has to do with the different steps that are needed before you get to the actual differentiation part with the donors, and that could perhaps influence it, or likely. But yeah, the way we have our biobank, it works already after 2 weeks of our liquid interface to have yeah, at least what you saw, the ciliary beating and all the cell types involved.
Wonderful. And then a follow-up question was, how long can you have the cells in culture for?
Well, we never tested how long, but we have done an experiment. We have published a paper with SARS-CoV-2 where we kept them on 5 weeks for our liquid interface. But I've also heard people doing it for months. I think, yeah, we also left them sometimes longer. I don't think they, once they are differentiated, I think they're pretty stable. I would expect them to be fine for weeks till months, but we never tested it. I mean, it costs money. So if you don't really have a purpose for it, we didn't test it, but I can really imagine that they can do quite a long time.
Very nice. Again, I want to thank you, Dr. Vander Dues, for your time today for PCLS. And I want to thank all of our attendees as well. So thank you so much, and I hope we'll follow up.
Yeah, I'm very sorry that my videos didn't work out, but I was very happy that I could share this work today. So again, thank you for the invite and for the questions.
Thank you so much, and have a great week, everyone.